Hello students, welcome to EPG Pathshala. My name is Vidya Kothekar. I had been professor of biophysics at All India Institute of Medical Sciences, JP Institute of Information Technology Noida, and Dr. D.Y. Patil Biotechnology and Bioinformatics Institute and taught biophysics for more than 40 or 50 years. Today, we are going to discuss about a module, bases, sugars and nucleotides under the paper biomolecules and their interactions. DNA, the full form is deoxyribonucleic acid. The double helical structure of DNA is one of the greatest discovery in the 20th century. It was discovered by Francis Crick and James Watson in 1953, and it had a great economical impact. Why? Because the discovery of DNA gave birth to the great Human Genome Project, one of the greatest projects of the 20th century. It is called HGP, which took place almost around 40 years. The spending between 1990 to 2003 on this project was 3.8 billion US dollars. But this is nothing compared to the economic impact that the discovery of DNA structure gave. It was 796 billion US dollars. This is the latest report by Jonathan Max. Students, let us first see what you are going to learn in this module. First, I would like to enumerate the basic building blocks of nucleic acids. Next, I want to describe nomenclature and different tautomeric forms, occurrence and importance of nucleic acid bases, deoxyribose sugars and ribose sugar. Then I want to distinguish between nucleotides and nucleosides. I want to elaborate on conformation around the glycosylic bond and sugar pucker. We show in this picture the famous photograph of Watson and Crick along with their model of DNA of the size of a room. Introduction. All cells, whether eukaryotes or prokaryote, they have genetic information, and this genetic information is coded in the sequence of the DNA molecule in terms of 4 nucleotide bases: adenine, guanine, thymine and cytosine. Chemical structure of DNA. The DNA molecule thus is made up of 4 nitrogenous bases: adenine, guanine, thymine and cytosine. They have phosphate and a sugar, deoxyribose sugar group. The polymer is built by attaching these bases to sugar phosphate backbone. DNA backbone. We show in this picture of DNA backbone given by Watson and Crick. It is shown schematically. The backbone is of alternate phosphate, sugar, phosphate, sugar, phosphate, sugar. The bases adenine, guanine, thymine, and cytosine, these are attached to the sugar units. DNA is a molecule of heredity. Why? Sorry, ये वीडियो कहीं था. DNA is the molecule of our heredity, and that's why it is the most important molecule in our life, because it connects us to our ancestors and different species, etc. The DNA molecule does this through the genetic information. This information is transferred from generations to generation by sequence of 4 nucleotides: adenine, thymine, guanine, and cytosine. How? The information is copied into a complementary sequence like a carbon copy, and then it is passed to the next cycle, then it is passed to the subsequent cycle. This is done by process of replication. The sequence of nucleic acid bases in DNA controls the sequence of amino acids in a protein chain. That is why it is so important. The DNA molecule is also very important because of its functions. The base sequence of DNA information is passed as 3-letter codon to ribosome through a process of transcription and translation by mRNA and tRNA, ribonucleic acids. The RNA also consists of heterocyclic bases, adenine, guanine, and cytosine. However, it does not have thymine, but in place of thymine, it has uracil. Again, the sugar in the ribonucleic acid is different. In DNA, we have deoxyribose sugar, while in RNA, we have ribose sugar. We show in this picture the nucleic acid bases. These are heterocyclic, aromatic, 5-6 member fused rings are purine. Adenine is a purine, so also is guanine. These have different functional groups attached to them, like adenine has NH2 group, guanine has NH2 group, and carbonyl group. There are 3 heterocyclic aromatic groups which are bases: uracil, thymine, and cytosine. These also have different functional groups attached to them. Like uracil has 2 carbonyl groups, so also thymine has 2 carbonyl groups, but thymine differs from uracil that it has extra methyl group attached to it, and cytosine has 1 NH2 group and 1 carbonyl group. Most important thing about the nucleic acid bases is a very recent study of meteorites by NASA showed the existence of organic compounds of life such as nucleobases in DNA and RNA, adenine, guanine, xanthine, hypoxanthine, 2,6-aminopurine, 6,8-diaminopurine in the outer space. Another interesting thing is this can also be formed in laboratory under outer space condition. Nucleobases have different tautomers. We show in this picture first the nomenclature for purines and pyrimidines like adenine and guanine, which are fused 5-6 aromatic rings. We start numbering from N1, C2, N3, C4, C5, C6. Amino group NH2, it connected to C6. N7, C8, N9. The glycosylic linkage in purines is through N9. Guanine is similar to adenine, only oxygen is connected to C6. The pyrimidines, cytosine and thymine, are numbered as N1, C2, N3, C4, C5, and C6. The linkage to the sugar phosphate backbone is through N1. NH2 is connected at C4 and carbonyl oxygen connected to C2. Thymine has same numbering as cytosine, N1, C2, N3, C4, C5 and C6, whereas two carbonyl groups are connected at C2 and C4, and there is a methyl group which is connected at C5. DNA bases also have minor tautomers. Like normally, adenine and cytosine are present in their amino form, but this can also go into imino form. In this case, one of the hydrogen at the 6th position shifts to N1 position in case of adenine. Similarly, in case of cytosine, one of the hydrogens of 4th position shifts to 3rd position N3 and it goes to imino form. Guanine is normally present as lactam form, which has carbonyl oxygen. So also is thymine. But it can also be present in lactim form. In this case, there is a shift of hydrogen or a proton from N1 to the carbonyl group and it goes to lactam form. So also in case of thymine, the proton from N3H shifts to O4. Cytosine. Cytosine was discovered by Albert Glycosyl and Albert Newman in 1894 when it was hydrolyzed from calf thymus. The structure was proposed in the same year. It has been found to have a great use in quantum computing. We show in this picture a cardboard model of cytosine, and for your information, Watson Crick built their original structure using the cardboard models. In this model or the picture, we show the nitrogen with the hydrogen atoms in blue color and the two hydrogens attached to a nitrogen 4 by red color. The nitrogen at the N3 position and oxygen at the O2 position, which are electron acceptors, are shown by green color, and the nitrogen N1, which is involved in the linkage with the linkage with the sugar phosphate backbone is shown in yellow color. Guanine. Again we show here cardboard model of guanine with electron donors shown in red color, which are the hydrogen attached to nitrogen atoms, and electron acceptors O6, N7 and N3 by green color. N9, which is glycosylic linkage, is shown by yellow color. The first isolation of guanine was reported in 1844 from excreta of a seabird known as guano, used as a fertilizer and named as guanine in 1846. The structure was determined by Fischer, who showed that it can be converted into uric acid. Adenine is quite similar to guanine, with the only difference of having a amino group at the 6th position with 2 protons H61, H62 shown in red color. Again we show the cardboard model of adenine. Thymine. Thymine is quite similar to cytosine, but thymine has a methyl group here shown in light blue color in the cardboard model, and these methyl protons are also quite important in recognition of proteins and drugs and other molecules. There is another proton at C6, the electron donor shown by red and the oxygens by green and glycosylic linkage by yellow. Methyl group is attached to the position 5. The amino group at 4th position is replaced by carbonyl oxygen, and N3 is protonated and linkage to sugar is through N1. Uracil. Uracil again is quite similar to thymine. The only difference is it does not have methyl group at position 5. We are going to show here a cardboard model. Ribose sugar. We show in this picture the ribose sugar. The sugars found in nucleic acids are pentose sugars with 5 carbon atoms. Ribose is found in RNA and is a normal sugar with 1 oxygen attached to each carbon atom. You can see here the nomenclature, that is C1 dash with OH, C2 dash with OH, C3 dash with OH, C4 dash with OH. Deoxyribose sugar is quite similar to ribose sugar with only one difference, that it lacks 1 oxygen and hence gets the name deoxy during glycosylation. There is a loss of 1 water molecule when it connects to nucleobases. Sugar puckering. The 4 carbon atoms C1 dash, C2 dash, C3 dash, C4 dash and oxygen O4 are not in the same plane. The sugar rings are puckered. If we consider a plane through C1 dash, O4 dash and so forth, 4 dash, the base nitrogen and the C5 dash form a boat-like structure. C3 dash and C2 dash, which are the 2 other atoms left, can lie either on the same side, and this is called an envelope conformation, or this can lie on opposite side which is called twist conformation. We show in this picture the two different sugars, Cytidine and Cytidine. Just by looking at them, can you make out the difference? What is the structural difference between the two? To clarify it further, we find an easier way, which is shown in the next slide. Just imagine a plane O4-, C1-, and C4-, and we color the two C2- and C3- by green and yellow color. C3- is colored green, C2- is colored yellow. If C3- is up, then it is C3-. If C3- is other side, it is C2-. This is shown in the next slide. So we show in this that is C2- yellow up, which is C2- endo sugar pucker, and C3- up, that is C3- endo sugar pucker. These are also called C2- exo or C3- exo. What are nucleosides? Nucleosides are N-glycosides of ribose and deoxyribose. Adenosine has ribose sugar, whereas deoxyadenosine has deoxyribose sugar. And we show the difference between the two. In the deoxyribose sugar, you do not have a hydroxy group at the second position. What is the difference between cytidine and deoxycytidine? The same. In the case of cytidine, the cytosine is connected to ribose sugar, whereas in deoxycytidine, it is connected to deoxyribose sugar. Pseudouridine and uridine. In the case of pseudouridine, the sugar is connected to C5 and N1H can now participate in H bonding. These occur in case of tRNA structure, and pseudouridine is different than uridine. Nucleotides. Nucleotides are defined as compounds where nucleoside is connected to a phosphate group. The building blocks of DNA and RNA are nucleotides. Nucleotides are generally considered as nucleoside monophosphates. However, nucleotide diphosphate, nucleoside triphosphate are also called nucleotides only. Conformation about the glycosyl bond. Relative to the sugar moiety, the bases can adopt two-bone orientation around glycosyl link. These are called syn and anti. In anti-conformation, the phosphate group is turned away from the bases. In the syn orientation, phosphate oxygen is near N3 of purines. This is shown in the next slide. We show in this figure Guanosyn-Syn in the syn conformation. You can see that N3 of the guanosine is quite close to the phosphate group. What is the difference with the anti-conformation? That we shall show in the next slide. You can see adenosine anti. Here, the phosphate group is turned far away from the N3. We show here N3 and show the phosphate group. How is glycosyl bond formed? OH from aromatic portion of the sugars and H from nucleic acid bases, that is N1H from pyrimidines and N9H from purines, combine to form a water molecule, and this is how the glycosylic linkage is formed. So students, what have we learned today? We have learned first the chemical structure, nomenclature for nucleic acid bases, ribose sugar and deoxyribose sugar. Nucleic acid bases are also called nucleobases. We use either a 3-letter code ADE, GUA, THY, CYT, or 1-letter code A, G, T, and C. Then we saw major and minor tautomers and the difference in the chemical structure between major and minor tautomers and importance of their coexistence. Third, we saw what is sugar pucker. It's a pucked ring. It's not a planar ring. Something goes up or it goes down. And we saw how the sugar pucker is nomenclature for the sugar picker, that is for whether it is a C2-endo, C3-endo, C2-exo or C3-exo sugar pucker. Then we saw the mechanism of glycosyl bond formation, and we also saw the different conformations, syn and anti, about the glycosyl bond. Then we saw the difference between nucleosides and nucleotides. Actually, many people get confused. Nucleic acid bases, nucleosides, and nucleotides. So, nucleic acid bases are only aromatic nitrogen containing fuse 5, 6 or 6-member rings with side groups, whereas nucleosides have sugar group attached to the bases and nucleotides have a phosphate and sugar group attached to the nucleic acid bases. Then we saw conformation around the glycosyl bond and explained the nomenclatures for different sugar puckers. I love DNA for many things. I love DNA because it connects me to my ancestors. It is another reason. You might laugh at it. I love DNA for its beauty, for its curvatures, for its shapes. I like to hang a picture of DNA in my drawing room. And again, I thank you all for your patience and going through this module presentation. Thank you very much.